Cell membranes! Cell Membranes by Laci, Cahill, Katie Randorf, Emily Hebert, and Seanna Pratt. All living cells are composed of a variety of things. There are organelles that provide the cell with energy, carry genetic information, store food, waste, and more!
The lipid bilayer acts as a barrier that keeps the stuff on the inside separate from the outside. There are two important regions of this bilayer, the heads and the tails. Each molecule contains a hydrophilic head region that loves water and a hydrophobic region that doesn't like water. The hydrophilic heads are on the outside of the bilayer and the hydrophobic tails are on the inside.
The phospholipid bilayer is amphipathic.
Huh?
All amphipathic means is that the lipid is both hydrophilic and hydrophobic.
Oh!
The phospholipid bilayer is not only composed of phospholipids, there are also a variety of proteins inserted asymmetrically. There is no exact arrangement. These proteins can be amphipathic too.
When we think of the term fluid in our everyday lives, we might think of something liquid, like water. And maybe mosaic tiles for the term mosaic. While these are accurate connotations, that's not exactly what we mean in regards to cell membranes.
What we mean by fluid is that the phospholipids and proteins are able to move laterally. And all the term mosaic means is that the bilayer is a combination of different things. Think of the cell like a VIP room. The cell membrane acts as a bouncer that only lets the certain molecules go through. The only molecules that are allowed in are the ones that diffuse most easily. Those are the small, uncharged, and nonpolar molecules.
This doesn't mean cell membranes are impermeable to other molecules. It just means they move a lot more slowly, or they need a little help from membranous proteins.
Many factors can affect the permeability of a membrane, including the temperature, composition, structure of the bilayer, the number of sterile molecules in the membrane, and the feature of the diffusing molecule.
Membrane permeability increases as temperature increases. It's important to note that after a certain temperature, membrane stabilization will occur.
Temperature is a crucial part in making sure proteins in the bilayer are functioning properly. Too much heat will cause denaturation, and the protein will lose its three-dimensional shape and function. On the other hand, cold temperature will cause the cells to expand and then burst.
Composition and structure can vary widely in bilayers and will affect the permeability of the membrane. A lipid bilayer containing no unsaturated fatty acids will have a lower permeability and be less fluid.
The tail-to-tail interaction between molecules increases as the hydrocarbon tail length also increases. This means that fluidity of the membrane will decrease, as does the permeability. The presence of sterols diminishes membrane permeability because they prevent solidification of the membrane at lower temperatures.
You may have heard people refer to certain fats as good and bad. What classifies a fat as either of these has to do with the number of saturated fatty acids in the lipids of the membrane. Coconut oil is loaded with saturated fatty acids. It is 92% saturated, whereas extra virgin olive oil is a mere 8% saturated. We can observe the differences just by looking at them. Because coconut oil is so saturated, it is solid at room temperature, whereas olive oil is a liquid because it's primarily composed of unsaturated fats. An interesting application with oils and membranes was an experiment that studied the effect of membrane filtration on the taste of olive oil. They found that they are able to effectively process an extra virgin olive oil without compromising the composition. This processing allows them to remove various compounds that can affect the flavor, thus giving it the best tasting olive oil to use. Cell membranes are essential for life. There are numerous components that make up its structure and even more that affect how well it is able to function. It is important to know how they work in order to understand how life is sustained.